Hello everyone, welcome to Smita Kiran Biology classes. I hope you are all good. In this class, let us discuss the process of DNA replication. You know that already Watson and Crick has proposed DNA double helical structure. Almost at the same time, that is immediately after the proposition of DNA double helix, same two people, Watson and Crick, also proposed the scheme for the process of DNA replication. That too, semi-conservative method of DNA replication. And the definition or statement for semi-conservative DNA replication is explained by their hypothesis. That is, it has not escaped our notice that a specific pairing we have postulated, that is, purines will always base pairs with pyrimidine, was the hypothesis proposed by Watson and Crick during DNA double helical structure. Immediately suggests a possible copying mechanism for the genetic material, that is, the information present in one strand will be copied to the other strand during the process of DNA replication. And that was explained by semi-conservative DNA replication. Now let us understand what is actually meant by semi-conservative DNA replication. Semi-conservative DNA replication states that two strands could separate during the process of DNA replication and they act as a template, and new complementary strand will be synthesized for those two DNA template strand. Here the which one I have written in black is a parental or template strand, and red color I have written is newly synthesized strand. And always remember, newly synthesized strand is complementary to the parental strand. So this is called, when there are two DNA molecules synthesized, it contains one purely a parental strand and another contain a newly synthesized strand. That is called semi-conservative DNA replication. You remember that was proposed by Watson and Crick. And there was other two methods of DNA replication proposed by other scientists. The one among them is conservative DNA replication. It states that during the process of replication, there is synthesis of two DNA molecules. And among those two DNA molecules, one is original DNA that resembles the original DNA, and another is synthesized will completely be new. But this was not accepted because of there was no enough experimental proof for that. And another type of DNA replication model was dispersive DNA replication. Here also there is synthesis of two DNA molecules, but within these two DNA molecules there is a patches of parental DNA and there are a patches of newly synthesized DNA. And remember, conservative and dispersive DNA replication models were not accepted because there was no enough experimental proof for that. But the well accepted and whatever we are studying about replication is only about semi-conservative DNA replication, because it was proved with a experimental proof, and that experimental proof was provided by two scientists, Meselson and Stahl. Okay, you have to remember here who proposed semi-conservative. The answer will be Watson and Crick. Okay, but they did not prove it experimentally, but DNA replication, that is semi-conservative DNA replication, was experimentally proven by Meselson and Stahl. Now onwards we will be discussing in detail about Meselson and Stahl experimental, that is the experimental proof for semi-conservative DNA replication. Semi-conservative DNA replication was proved by two scientists, namely Matthew Meselson and Franklin Stahl, in the year 1958. So you have to remember the two scientists' name, because in this chapter you will be having many scientists and their contributions. So don't get confused with that. Matthew Meselson and Franklin Stahl in 1958 have conducted an experiment to prove DNA replication, that is semi-conservative method of DNA replication, and this experiment is popularly known as Meselson and Stahl experiment. During the experiment, they took bacteria called Escherichia coli, and Escherichia coli multiply for every 20 minutes. So that is the generation time. Every living organism has its own generation time. Likewise, bacteria Escherichia coli will generate its new generation within 20 minutes. And Escherichia coli have a genetic material, of course, which is DNA, and that DNA is not present in the nucleus. It is present as nucleoid. You have studied in packaging. Now, the DNA is consisting of nucleotides. So we will say DNA is a polymer of nucleotides, or nucleotides are the monomeric units of DNA. And each nucleotide has three components: nitrogenous base, that is purines and pyrimidines, phosphate group, that is, and the pentose sugar, which is deoxyribose sugar in case of DNA. Now, nitrogenous bases consisting of, as the name suggests, nitrogen. It contains nitrogen. Naturally, the nitrogen present in DNA is N14, which is a lighter isotope. Okay. Now, let us explain experiment in 3 steps. In textbook also they have explained the experiment in 3 steps. Let us consider the first step. In first step, what they did is, Escherichia coli bacteria was allowed to grow in a medium. You have to remember nutrient medium. Nutrient medium means it will have all possible nutrients, carbohydrates, proteins, nitrogen supplement, everything. Nitrogen supplement they given was ammonium chloride, and that ammonium chloride contain a heavy isotope, that is N15. But the naturally present was N14, but what they supplied is N15. So when this was grown for many generation, they grow the Escherichia coli bacteria only by providing nitrogen 15 isotope in the form of NH4Cl. Now all the DNA, that is all the cells which are produced, will only have nitrogen 15. Sorry, I have written here as N14, so that's not N14, that is N15. So which is a heavy DNA. Okay, so which one is having N14 is a lighter DNA, which one is having N15 is heavy DNA. So don't get confused here. I have written the N14, it is not N14, it is N15. Now, these two, lighter and heavier DNA, can be distinguished only based on their densities. So isotope here, that doesn't mean radioactive. N14 and N15 are not radioactive isotopes. They are density, they can be differentiated only based on their density. So when the lighter DNA is subjected to cesium chloride density gradient centrifugation, there was a formation of band above, that is surface, whereas the heavy DNA was subjected to density gradient centrifugation. The band was formed at the bottom, which suggests it is a heavy DNA. At second step, what they did is, the Escherichia coli bacteria was transferred on to a natural nitrogen containing medium. That is, here there was a supplement of ammonium chloride, which is having N14, which is a natural isotope. Now, when they growed it for few generations, so you have to remember, they did not allow it to grow for many generations. Here they considered the DNA molecules to be grown for a few generations. At definite intervals of time, they allowed to grow. So here what I am explaining, they took the Escherichia coli bacteria, which is grown on N15 medium for many generations. Now what its DNA contain? Only N15. So now the cells are allowed to grow on NH4Cl, that is nitrogen 14 containing medium, for many generations. So that is, if it is allowed to grow for 20 minutes, that will be first generation. After 40 minutes, it will be second generation. After 60 minutes, it will be third generation, and so on. Okay, so this was what they did. Now, they accordingly, that is periodically, that is after 20 minutes, after 40 minutes, after 60 minutes, they analyze the DNA for their separation in density gradient centrifugation. How analysis was done, let us discuss in the third step. Before starting this, I'll recall once they grown bacteria in a nitrogen 15 medium, they obtain nitrogen 15 containing DNA. Okay, so or nitrogen 15 incorporated Escherichia coli cells, and those cells are grown in a medium which having nitrogen 14 isotope. When they isolated the DNA from Escherichia coli which were grown on N15 medium, the band was formed at the bottom because two DNA molecules or two DNA strands are having N15 only. If the cells are allowed to grow on nitrogen 14 medium and the cells are analyzed for the DNA, after 20 minutes the band was formed at the middle because the two DNA strands which are templates are having N15, but the newly synthesized DNA strands will have N15 because the provided medium is having only N15. There is no other chance. Now this is called a hybrid DNA, and hybrid DNA will always form a band at the middle. Now after 40 minutes the same cells are analyzed for their DNA in cesium chloride density gradient centrifugation. There the bands were formed of two types. One was lighter band which is formed above, and another band is formed at the middle. Of course it will be a hybrid. Reason for this is in previous one whatever they analyzed, there are four template strands, among which two strands are having N15 and two strands are having N14. But the four strands which are newly synthesized, all of them are having only N14 because the provided medium is having only N15. Now there is formation of 4 DNA molecules after 40 minutes. Among them, 2 are hybrid DNA molecules and 2 are lighter DNA molecules. This is clear from the above evidence, that is after separation. That means equal amount of hybrid DNA and the lighter DNA molecules are formed. This is purely based on the template. Now let us see what happened after 60 minutes. Here I have written 30, I made it 40, so don't consider it. After 60 minutes, if the cells are isolated with the DNA and DNA is subjected to density gradient centrifugation, now there was formation of a larger band above, that is lighter band. Lighter band was having more DNA, whereas the middle, that is hybrid band, is having only little amount of DNA. Reason for this is in previous, that is after 40 minutes, there are 4 DNA molecules formed, which means 8 template. Among those 8 templates, 6 templates are having N14 and only 2 templates are having N15. So in diagram it is clear. Now N15 containing DNA will form a band at the middle. And how many DNA are there? There is only 2 DNA molecules. So that is forming a thinner band, whereas lighter DNA molecules will be forming a thicker band. That means lighter DNA are more, whereas hybrid DNA are less. And after 80 minutes, that is question is asked in NCERT text, after 80 minutes there is formation of 16 DNA molecules, because in previous there are 16 templates. Now among them 14 templates are having N14, 2 templates are having N15. So lighter DNA will be 14 molecules and the intermediate, that is hybrid DNA, are 2. So from this it is experimentally very proven that the DNA replication is semi-conservative. Now Meselson and Stahl experiment was conducted using Escherichia coli bacteria, which is a prokaryote, and it contained both genomic DNA as well plasmid DNA. Now Taylor and colleagues in 1958 have conducted an experiment to prove whether the chromosomal DNA will replicate in the semi-conservative method or any other method. Experimentally, the similar kind of experiment like Meselson and Stahl experiment was conducted, but here they used a plant, Vicia faba, or it is called broad bean or faba bean. To prove this, they used radioactive thymidine, which is a nucleotide. They used radioactive thymidine. Here they did not use any elements. Here they used a molecule, that is radioactive thymidine, to detect the distribution of DNA in a chromosomes. So here, after the experiment, they proved that the DNA replication, that is the chromosomal DNA replication, is also follows the same, that is semi-conservative DNA replication itself. Okay, so by this it is very clear that any DNA molecule in an organism, whether it may be a genomic DNA or a plasmid DNA, anything will always replicate by semi-conservative method. That means the DNA molecule will retain one parental strand and it will synthesize as a newly synthesized strand. And remember, this topic is very important for board exams and as well as for theory exams. In next video, I will explain the process of DNA replication.